And I'm going to start out this evening, for me it's evening, but to welcome everybody from around the world to the Siwalik Seminar Series. And Irfan in Calgary is a little bit under the weather, so he might be listening, but he's not joining us actively. I'm just going to point out that in the next couple of weeks, we've got seminars scheduled for Parth Chandra from the Indian Institute of Science Education and Research. That's going to be next week, and he's going to be talking about some of the quest for hominid fossils in the Siwaliks. The following week, we're going to have Lisa Taux from Santa Cruz talking about magnetics, paleomagnetics. And the following talk is going to be by Peter Zeitler from Lehigh University. And he'll talk about provenance patterns and mineralogy of the sediments in some of the Siwalik rocks. So and we've got, after Peter's talk, we've got 4 or 5 people lined up. So we'll be running the seminar through June and into July. And I hope that you'll all continue to join us. And we're extremely grateful to Mukhtiar and his people in Quetta. And they are systematically recording these presentations and putting them on the YouTube channel. So you can all access the information on YouTube. And I see we've got several of our previous speakers. I just want to thank Kay Berensmeyer for being up in the middle of the night, and Catherine Badgley being up in the middle of the night, and Andrew Meggs is here, not so much the middle of the night, and some other people. So thank you all for being here. I'm going to introduce our speaker tonight. Jason Head is with us from Cambridge University in England, and Jason has worked on the terrestrial and aquatic reptile faunas from the Siwaliks, and is interested in the relationship between the reptilian fauna and the paleo environments and paleogeography of the Siwaliks and I might say the Himalayan foreland basin. And we've had a nice little chat here just before the session, and Jason has crossed paths with areas that many of us have worked in Wyoming as well as in Kenya. And so I'm delighted to welcome to the seminar series here. And Jason will give you the floor.
Well, thank you very much, Bob. So yeah, I'm very happy to be here. It is very early in the morning UK time. It is 5:30 a.m. I actually miscalculated when I would be giving the seminar. I thought it was going to be at 4:30, so I've been up since 3 a.m. And so while I can now say that I am stable enough in my career that I can publicly admit that I have occasionally fallen asleep during a scientific talk, today may be the first time I fall asleep giving a scientific talk. But I will do my best, and I am very, very happy to have been invited and to participate. And the reason I am is, of course, because the Siwalik was my first real experience into Cenozoic paleontology and into kind of dynamic field work where you're really combining a lot of field geology and sedimentology and stratigraphy with a vertebrate fossil record. And so what I will do is see, I think everyone can see that. And so let's go view. Can everyone see that?
Yeah, it's looking good.
Fantastic. So what we're looking at, whoops, stop that. In this image is the view kind of across the Dokbatan Formation to the town of Padri, which is just slightly visible on the horizon at sunset. This is my first field experience in 1996 in the Siwalik. And my interest is in reptile fossils and what they can kind of tell us about both the evolution of modern ecosystems that we have today and kind of how those ecosystems have evolved in response to climate change, to tectonic histories. I'm interested in things like biogeographic patterns. And so what I'm going to talk to you about is some of the work that I've done with the Siwalik reptile record, some of the work I still hope to do, and kind of explain a little bit about what the nature of the record is like. And so to kind of contextualize my interest in the Siwalk Group within a larger framework, I'm interested in really understanding the evolution of modern tropical diversity in reptiles. And for reptiles as being poikilothermic or cold-blooded, which is a terrible term, but is one I think everyone generally understands, is animals whose metabolic rates and ecologies are very much dependent on the ambient temperatures in the environment that they live. The tropics are extremely important, and the highest biodiversity of reptiles today is at the lowest latitudes. So here is just kind of a simple schematic of southern Asia down through Australia. And what I'm interested in is what the Siwalak Group can tell us about the evolution of some of the kind of biogeographic provinces, some of the biomes that we see kind of along this stretch. And so in this map, you can see, of course, a couple of the famous biogeographic lines that we have between Australia and running through Southeast Asia, such as Wallace's classic line, which separates the Greater and Lesser Sundas along the Lombok Strait. There's Huxley's line, which is a modification of this, that incorporates part of the Philippines into the Greater Sundas, and then Lydeker's line, which separates the kind of Australo-New Guinean component of Australasia from the Lesser Sundas as well. And so from those patterns and from a lot of exploration, of course, we recognize towards the bottom here, we recognize several biogeographic provinces. There is the Sahel Shelf province, which has a fairly endemic fauna, which is in the case of reptiles, and this is the perspective I'll be talking about today. We have things like the pig-nosed turtle here, carettochelyid and sculpta, which is endemic to that part of the world and occurs nowhere else. We have Wallacea here, which is, of course, part of the Lesser Sundas, which very famously has taxa such as the giant lizards, the Komodo dragons. And then we have the Greater Sundas, which are a component, along with South Asia, of the larger Indo-Malayan biotic province. But actually, the oceanic component here, the island chains, which form part of the submerged platform of Southern Asia, has a series of endemic faunas, such as this false gharial right up here, and a diversity of mammals and birds that we'd all recognize. One of the kind of endemic with some fuzzy edges to it components is this of the Indo-Malayan biotic province is the South Asian province, which I've outlined here, which of course includes kind of what was classically known as the Indian subcontinent, plus variably bits of Myanmar and Thailand, depending on the taxa you're looking at. And these have some kind of classically endemic taxa as well, such as the long-nosed and critically endangered Indian gharial. So what I'm interested in looking at these provinces is kind of what's the timing of their assembly, or what's the directionality of their assembly, and what the fossil record can tell us about that. Now, we have some good sense from other data sources about kind of when we get some of these faunas, these kind of insular endemic faunas evolving. So this was a paper that actually looked at evolutionary relationships determined by genetic data from a wide group of animals as well as plants between what these authors referred to as mainland Asia and the Indian subcontinent. And what they were interested in doing is actually mapping out phylogenetic relationships, or evolutionary relationships of taxa that cross both of those areas, and then actually looking at when the common ancestry between those two groups would have happened based on calibrated phylogenetic trees, and then using those determinations to actually model when the pattern of immigration both from the Indian subcontinent into mainland Asia and from mainland Asia into the Indian subcontinent would have occurred. And so what you're looking at is kind of these frequency distributions that you see. For example, reptiles have the highest pattern of interchange between mainland Asia and, again, what they refer to as the subcontinent, between 20 and 15 million years. Fishes, roughly about the same, although they have two peaks. Mammals, slightly younger at just under 10 million years, but with a peak rising up from about 15 million years to 10. And this paper kind of finds a generally similar pattern that they calculated really had its highest peak at around 14 million years, which is the middle Miocene climatic optimum. And so these authors conclude that what we have in terms of the evolution of the kind of modern South Asian biogeographic province is something that happens in the middle Miocene, where you have the immigration of groups from Greater Asia into this area, basically evolving into the ecosystems that we have today. So these are two of the larger frameworks that I'm interested in how the Siwalak record might inform us. And of course, I'm also interested in how reptiles respond to climate change through time. As I mentioned, reptiles are poikilothermic, or cold-blooded, so they're very good and very sensitive indicators of environmental and climate change. From Catherine's talk and from some of the other talks, you've already seen some of these images here, Julius's amazing reconstructions of the middle Miocene here with Y496 at about 12.4 million years. And then right here at Y581, a younger locality in the 7 million year range. And of course, the environmental differences between these two, where the image at the bottom shows a fairly closed, canopied, wet, forested environment. But the one at the top is an more open environment with grasslands. And of course, this corresponds to the environmental changes that we see both in terms of the vertebrate biomes, or excuse me, vertebrate faunas, as well as recorded in the geochemical signals, both in pedogenic and in biogenic reservoirs. So from the top here, of course, this is the pattern that we see in carbon-13, showing that very sharp excursion starting just under 8 million years. That demonstrates a transition from C4-dominated photosynthesis to from C3-dominated photosynthesis to C4-dominated photosynthesis, and then this transition from closed forest to open grasslands. And then the pattern what we see in oxygen, which shows a transition, probably it's more diffuse, but roughly at around 10 million years, where we start to see oxygen isotope ratios that are more consistent with seasonal precipitation and possibly the inception of Asian monsoons. And so we have this very good, very well-constrained environmental transition recorded on the Potwar Plateau. One of the things I've been very interested to see is how the reptiles respond to it. So for these questions, one of the things, if we're interested in reptiles, is we have to look at the South Asian reptile record, where it is and how old it is. And there's an asterisk next to South Asian. And that's because for this image, I'm including a couple of records from areas that have kind of traditionally been separated from that. That includes a record described from Afghanistan, as well as a couple of records from Myanmar and from Thailand, which are, again, slightly outside of this biogeographic realm, but are important for informing on a lot of the kind of herpetofaunal components. And then what you can see is the age range for these areas ordinated by the oldest estimated ages for them. And this thick dark line is, of course, the Potwar sequence, which goes from about 18.25, give or take, all the way up to about 3.5. That's, of course, the Potwar sequence writ large. The reptile record that I've been able to sample or examine is actually not represented through this entire interval. And so what we know about the history of reptiles from one of the areas that has their highest biodiversity today and has undoubtedly been an important engine for generating that diversity is from this series of kind of 19 specific collecting areas. And you'll notice that, again, most of them are of either poorly resolved in terms of their temporal distribution or actually fairly short temporal ranges. And because of that, the Potwar record, I think, is kind of inordinately important for looking at long-term change. And where some of this comes from, just a little bit of the background of the history of reptile collecting especially in the Potwar sequence, or in the Siwalk writ large sequence. So the first collections, European collections, I should say, from the Potwar Plateau were made by this gentleman here, standing the third from the left. That's William Theobald from the Geological Survey of India. He visited the Punjab and the Potwar and made collections of fossil mammals and fossil reptiles, including a turtle shell from an area that he identified as Osnat, which I presume is actually the village of Hasnat today. And so Theobald collected some of this material. He described only a very little bit of it, and many years after he collected it did he finally actually get around to describing it. And so there's only a little bit early on of collection from the Potwar region. Most of the Siwalk writ large reptile studies were initially done by these two gentlemen here, Hugh Faulkner and Proby Cotley. Faulkner and Cotley, of course, were working more in the upper Siwalk, in the Pliocene region in India, around Chandigarh. And they described the reptiles as kind of a component of their larger studies. They actually did a fairly thorough job of illustrating and figuring all the reptile material they found. And then both Theobald and the Faulkner and Cotley materials were more thoroughly documented by this gentleman to the right. This is Richard Lydecker, who I was rather amused recently to discover actually graduated from the department within which I work at the moment. And what Lydecker did is basically go through these previous collections, describe all of this material, and he really kind of provided, especially some of the first systematic frameworks for some of the reptiles that we've come to know and understand today. Following Leidecker's work, most of the reptile studies were, again, part kind of tacked onto larger mammal studies. If you saw John Barry's talk, or if you watched John Barry's talk from this section on YouTube, especially things of Barnard Brown's expedition, the Yale University expeditions, collected materials, and some of the reptile materials described in one or two kind of small to medium monographs. It's important to recognize Richard Dame's expeditions from Munich. He collected a fair amount of material on the Potwar Plateau, especially snake material that was described by Robert Hofstadter in the 1960s. But beyond that, there hadn't been really much, especially in the Potwar region, that had been described at all. And so that's kind of the framework that I came into when I started graduate school. And just a little bit about that. So my interest originally in paleontology was in dinosaurs. And I ended up working with someone who actually is intimately involved with the early establishment of some of the big Siwalik projects. And that's this gentleman here at the bottom, Louis Jacobs, now professor emeritus at Southern Methodist University. Louis was a student of Ev Lindsay's, and they connected with the Dartmouth Peshawar research project. And Louis initiated a lot of the screen washing for micromammals in the Potwar Plateau. Starting in the late 70s. I think Larry Flynn might be giving a lecture at some point on going into greater detail on that. So I went to study with Louis, but it was many years after he had worked in the Siwalik, and he had actually transitioned on to working on dinosaurs at this point in North America. And so I went to go work with him to study dinosaurs from Texas for my PhD. And as I had been an undergraduate at the University of Michigan and very interested in kind of all things dinosaurs, but I also like reptiles. And as a result, Catherine Badgley, seen here on the left, actually gave me a couple of fossil turtles from the Siwalik to study and prepare. And I studied them and tried to understand them, but this was a very deep pool to jump into because turtle systematics are very touchy and difficult to discern and disentangle, especially when it comes to cranial anatomy of this particular group. But there was something that had happened because of my work with Catherine, which is soon after I got to Southern Methodist to work on dinosaurs, I received an email, and I'm paraphrasing with "don't study dinosaurs," even though it's in quotes, and I can't exactly remember the specific wording, but I'm pretty sure it starts with "don't study dinosaurs." And the email effectively said, "Don't study dinosaurs, too many people work on dinosaurs. I have this great fossil record of reptiles from the Miocene of Pakistan. You should work on them, and if you work on them, you can come join us in the fieldwork in Pakistan." And that email was from John Barry, who had clearly talked to Catherine. And so there's a picture of John along with Colin Jake and Will Downs in 2000. So my first real experience with Cenozoic paleontology came from Pakistan. And prior to that, I had only done some dinosaur quarry work in Colorado and Wyoming, and a little bit of work in the Triassic of Arizona. And my degree, my background at this point was in biology. I knew very little geology. So I went to Pakistan as my first international field work. As a matter of fact, outside from Canada, it was the first country I'd visited outside of the United States. And Canada is a bit of a cheat because I was actually born very close to the US-Canadian border. So I arrived in the Siwalak Group in 1996 to study the reptiles, and I was very excited, very confused. And my hope was to start to do with the reptile record what everyone else had been doing for 20 plus years with the mammal record. And I showed up with these kind of mental images of building this incredibly well-resolved pattern of first occurrence and last occurrence among the reptiles, and thinking about diversity changes and evolutionary rates and evolutionary transitions. And I was thinking about this because I didn't actually know that much about what the nature of the reptile fossil record is like. This is a figure from a paper that Kay and John wrote in 2005, and it is basically a biostratigraphic survey at different collecting levels from 10.4 million years to 7.3 million years. And what the chart shows is the relative percentage of specimens recovered for mammals, reptiles, and fish as a percentage of NISP, which stands for number of individual specimens. So my mental image of what the reptile record would be like and the resolution of the reptile record was quickly dashed by the reality of what you see in this figure, which is that if you go out and in the Siwalik and you walk around, most of what you're going to find in terms of vertebrate fossils are mammals, and reptiles are not represented nearly as well. I would say that, you know, if you look at this chart, it's averages somewhere around 30% of the record is going to be fossil reptiles if you go out and look for stuff. And there's a couple of reasons for this. The first is one of the reasons that is a reason that Catherine mentioned in her talk, which is that the mammal fauna of the Siwalik is large. There is a high diversity of megafauna, many large-bodied species, and so that image that you see over there with the pick, which are two large, I presume, artiodactyl vertebrae, a cervical on the left and a dorsal vertebra on the right. These large mammals make it into the fossil record with very high frequency. So you find a lot of large mammal bone. The majority of the reptile record consists of animals that are much smaller. And also the nature of the mammal fossil record is that a lot of it consists of teeth. And mammal teeth are a really important component to understanding the history of vertebrate life, because mammal teeth are unique in that they're very taphonomically durable. They're ecologically kind of very precise in what they can tell you. They're phylogenetically precise. They can tell you what the animal is. And that's because they're composed of this very thick prismatic enamel that is very resistant to the kind of physical and chemical destruction that will remove other mineralized tissue types from the fossil record. The mammal teeth are also very complex. This complexity is, of course, correlated with dietary specialization, and that complexity also is very useful for identifying different species. And I should also point out that, of course, mammal teeth are, because of the mineralogical composition of the hydroxyapatite of their teeth, they're also an excellent reservoir for stable isotopic data. So the fossil record in the Siwalik is much more biased both biologically towards mammals. The reptile record is not nearly as dense, but also reptiles themselves lack some of the tissue systems that make for easy understanding of things like taxonomic identity, ecology sometimes, or body mass. So I distinctly remember being in a little bit of shock when I got there and kind of found this mosaic of kind of turtle shell fragments, and then had to kind of figure out what I could say and what I could do with them. And the answer is actually you can really do a lot with this record. It wasn't what I initially hoped it would be, and it's taken me about 21 years to kind of develop the ability to do what I think are some interesting things with it. But you can make it work for you. And so what I'm going to do now is kind of introduce you all to what we can find in terms of diagnostic material from the record, and then I'll talk a little bit about what we can say from it. And so this is just an example of some of the fossils that we get. Some teeth. So what you're looking at here on the left are a series of jaw elements of lizards in multiple views. So on the upper left here, for example, you can see this is a multicuspid tooth on an upper jaw fragment in 2 views. This is a lower jaw. This is in kind of medial view, so the looking out from the interior of the jaw, that's looking down on it, that's looking at it from the lateral side, and that's looking at it from the underside. And so these are different jaw fragments of different kinds of lizards. I mentioned that just a second ago that we lack the kind of taxonomic precision from the reptile fossil record based on teeth that we get from mammals, and that's true to the level of, say, a genus or a species. But for some kind of higher order groups, whether a lizard belongs to a particular very inclusive lineage or an inclusive clade, we can get some of that information. So it's not as though the record is completely useless. So for example, up at the top here, the dentition that you see in these jaws are what is known as acrodont. And that is a particular type of dentition that is characteristic for a large group of lizards here. These include things like, this is an animal, for example, like Uromastix, which is an animal that you would see on the Potwar Plateau today. It includes some forest-dwelling species and some rock-dwelling lizards as well. Probably everyone has run into a gecko at one time in their life, and so we actually have geckos here, this little fragment of a jaw. And then these are lower jaw fragments of a very, very diverse group of lizards known as skinks. All of this material shows a kind of taxonomically diverse, although again somewhat imprecise, collections of lizards that all have different kinds of ecologies. Some of these ecologies are for herbivory, most are for insectivory, and almost all of these groups have modern representatives in South Asia today. And in fact, one of the things that I'll point out as I go through the various reptile groups is that a lot of the reptile clades that we find in the Siwalak group are actually endemic to, if not the Potwar itself, to some of the major drainages that are fairly close to the Potwar in modern Pakistan. So we have a diversity of these very small lizards. And again, I'm very sleepy, so I can't remember if I've already just said whether or not, but the images that you're looking at, they look kind of weirdly drawn. These are actually three-dimensional reconstructions from CT scans of the fossils. And I use these whenever I can because they provide actually greater ease of visibility and sometimes greater accuracy of depth than a standard photograph. One of the group of lizards that is fairly common, is present today, but is an ecologically important component of just about every terrestrial tropical vertebrate assemblage in the old world, is the genus Varanus. These, of course, are monitor lizards. They're represented primarily by fairly large vertebrae. You can see here in 5 views. So these are parts of the backbone. So that, for example, that's just an anterior view, and that's a dorsal view from the side, bottom, and from the back. This, by the way, that specimen is an animal that's actually represents a very large animal with a body length, not quite at 2 meters, but probably, including the tip of the tail, fairly close. And varanids are important because they're what we generally refer to as hypercarnivorous. They're vertebrate predators. They are predators of small mammals and other squamates, squamates being lizards and snakes. So we find these animals throughout the Siwalak group. The most taxonomically diverse group that we get are the snakes. And so this is an image from a chapter that I have in the kind of upcoming Siwalak monograph, showing different groups of snakes that are represented by the occasional cranial fragment as well as some postcranial elements. And I'll talk a bit about the kind of the snakes that we find. And one of the reasons I'll talk about that is that's the group I focused the most of my attention on. They have the densest record of any of the Siwalak group reptiles, the most continuous record. And I do want to kind of tip my hat to the Siwalophiles, especially. I know John Barry has made it a point for many years to collect all the snake vertebrae he could find, because very early on he recognized that they could probably tell a very interesting story. And he was correct. And so some of the snake fossils that we get, for example, this is on the upper left here. That is in 3 views, a cranial bone of python. So what you're looking at is the maxilla bone. So this is the upper tooth-bearing bone of the jaw. So it's the upper jaw. And if you look at this lateral view here, this component of the bone, that corresponds to this area right here on this python skull. And so the eye orbit of the animal would be right about there. And the fossils of the python that we find in the Siwalak group are anatomically very similar in both morphology, but also in body size to living Asian pythons today. And so here are a couple of vertebrae that I described in 2005. And for those of you who might not be familiar with pythons, it's not everyone is probably a paleoherpetologist or a herpetologist. Of course, pythons are very large terrestrial and semi-aquatic snakes. They are predators of mammals. They are predators of other reptiles. This gives you kind of a sense of a python. This is a bit of a cheat. This is an African rock python simply because I didn't have a good photo of an Asian python. So we do find the occasional skull bone for snakes, but very rarely, which is unfortunate because skull bones, unlike many of the components of the anatomy of these animals, are fairly diagnostic. You can have some good confidence in the identity of the animal based on skull bones. But generally what we find for the fossil record of snakes is actually vertebrae for backbones. And the reason for that is illustrated by the CT scan of the snake Samophis on the right. What you will notice is that the skeleton of a snake is basically the vertebral column, the spine, if you will, with this kind of slightly interesting little bit at one end, which is the head, and then many, many, many repeating units of vertebrae and pairs of ribs, which have some kind of subtle shape differences along their axis, but all have fairly complex and, to more inclusive taxonomic groups, all are actually fairly diagnostic. So you can actually get some fairly robust identifications from snake vertebrae. So some of these include Animals that you might not want to find as you're walking around. So for example, here we have a vertebra of a large viperid, such as Russell's viper here, or a saw-scale viper. And this specimen has a lot of the kind of very characteristic diagnostic features of these animals. At the bottom is a series of articulated vertebrae. Again, these are CT scans. This vertebra or this fossil in actuality is encased in mudstone. And these vertebrae here kind of make a very precise identification, very difficult, but this specimen probably belongs to a very diverse group of snakes called the lapoids, which includes things like modern cobras. And then this specimen here is very interesting because it belongs to the genus Bungarus, or also known as the krait. And I'm really happy to have found these specimens because kraits are one of the few snake groups that have diagnostic characters that you can actually, of their vertebrae, that you can actually use to identify them to within the genus. And so these specimens, this specimen here, as well as several others, has a little kind of flap there, and there's a little bit of a flap there. And on other specimens, you can see that the vertebrae are actually fairly wide at the joints where they articulate with the vertebrae in front and behind, which give them almost an appearance that makes them almost look like a bat wing when they're actually complete and not broken. And the morphology of these kind of processes is actually very diagnostic within the group, and this actually allows us to kind of separate these fossils not only to place them within the genus Bungarus, but also allows them to actually place it within the species complex of living Bungarus species, which ends up becoming important for doing things like calibrating phylogenetic patterns based on molecular data. So there is actually a diversity of snakes within the Siwalak group that are basically procured from both surface collecting but also screen washing. And the increase in diversity, and we'll see this in a minute, or there is an increase in diversity through time, as I'll show in a minute. But the one animal I also want to point out is kind of very, as it turns out, very important for thinking about environments and ecology in the Siwalak group, and that's the snake Acrochordus. This animal is, through the course of my graduate career and professional career, I think every paleontologist has a favorite fossil. This is my favorite fossil. It's my favorite living animal as well, and it's because Acrochordus is the single most abundant reptile taxon in terms of numbers of localities that you find them, and number of specimens, at least for individuals, through the section. And it is a very interesting animal. This is a vertebrae of Acrochordus in 5 views. You're looking at it kind of in the front here, and again, top, side, bottom, and back. The reason that we find so much of this animal is that, and we know this from the fossils and based on depositional environments, and we know this also from its living related texts, is that Acrochordus is a fully aquatic snake. And where these animals occur, they generally occur in very high population densities. And so as a result, we have a very dense record from aquatic environments, which almost certainly indicates the origin of some of these specialized ecologies and high populations very early in their evolutionary history. It's a very interesting animal just on its own right. Like I said, it's a fully aquatic snake. They almost never leave the water. This one is called the little file snake. It is actually a shallow marine specialist that occurs along the beaches in southern India, or in mangroves in southern India today. These animals are primarily occur kind of in the equatorial Indonesia, and then down through into northern Australia and going up kind of into both southern South Asia and southern Southwest Asia as well. They're really interesting animals because they're fully aquatic, but also because of their habits and their habitats. They are specialized fish eaters. And so this is a specimen of an Australian species that has unfortunately expired from attempting to eat a catfish that was too big for it. And the way that these animals actually acquire the fish is that they actually constrict them. And if you've ever dealt with a live fish, that's a tricky thing to do. So they have this very kind of rough, coarse and loose skin that actually allows them to gain purchase on a fish. Of course, they're not constricting the fish to suffocate it. They're constricting it just to hold it while they can eat it very quickly. So this animal here is actually a really interesting environmental indicator because, again, the living species of Acrochordus inhabit very low altitude, very warm, wet environments, some river systems, some brackish water systems, and some slower water systems as well. Living Acrochordus gets to body sizes around just under 1 meter for the little file snake to just about 2 meters in the largest file snake today. One of the interesting things about the Siwalik Group Acrochordus is that it's actually considerably larger. It's about twice the size of living Acrochordus today. What we have here on the right is a chart for a paper that came out a couple of years ago that is extremely useful for reconstructing body sizes or body lengths, in this case, in snakes based on vertebral measurements. And so I'm just showing you on the regression relationship here at the lower area. This is for the width of the cotyle, which is that kind of circular spot here. Excuse me, that's the part of the central articulation of the vertebra in these animals, the ball and socket joint. And the width of that socket actually can predict the length of the animal. When you apply, for example, that relationship to this specimen here, you get a body length estimate of just about 3.5 meters. And this is not the largest specimen of Acrochordus by far. What's interesting about that is, again, the Siwalik Group Acrochordus is much larger than we see living Acrochordus today. Some of these animals can achieve body sizes that are equivalent to some of the aquatic boid snakes like anacondas that we see in the neotropics today. The other interesting thing about the Siwalik record of Acrochordus is that the animal is kind of ubiquitous in middle Miocene age sections in South Asia. So it occurs in the Siwalik Group in the Potwar Plateau. It occurs in middle Miocene sequences in Gujarat, the Siwalik sequence in Nepal, and going all the way down actually to some of the middle Miocene in Thailand as well. So we know that the past distribution of this animal actually is kind of spanning the large floodplains at the base of the emergent Himalaya all the way down to the coast during the Neogene. So those are the squamates, the lizards and snakes, and so they include terrestrial animals like most of the lizards. They include obviously aquatic animals such as Acrochordus, and then a mix of probably terrestrial and aquatic snakes and some of the smaller animals. Turtle diversity is very high in the Siwalik Group as well. There are some really interesting animals and some unexpected animals in there. So this is one or two or three known specimens of a carettochelyid or a pig-nosed turtle that we find in the Siwalik Group. And this element that you're looking at here corresponds to this element right there in the shell. This is an element known as a peripheral element. These line the margins of the shell. And what you're looking at is this is the belly portion, the underside of the turtle here. The hind legs would be coming out there and there, the front legs there and there. Carettochelyds are highly aquatic adapted animals. They really don't come out of the water, just like Acrochordus doesn't. And what's interesting is, of course, today they're completely restricted to the Sahel platform. So we're talking about Northern Australia and Southern New Guinea. More common animals include, and by more common I mean very common animals, are the soft-shelled turtles. And any Cenozoic paleontologist who works in terrestrial environments in North America or in Africa or in Asia will have run into these fossils of these animals, the soft-shelled turtles. They are aquatic specialists. Trionychoids, as they're technically known, are characteristic for having either completely lost or greatly reduced some of the marginal elements of the shell, of having reduced the bony components of the shell to make the shell more flexible for fast swimming underwater as well as burrowing. And so we actually have a very good fossil record of soft-shelled turtles from the Potwar section, including this little animal here, frankly adorable animal. This is Lysemys punctata, the Asian flap-shelled turtle. Lysemys is most closely related to a radiation of African turtles, and it has a very characteristic kind of ornamentation and patterning to the shell. So you can kind of see this surface sculpturing, this kind of punctate, hence the name punctata. And the elements that you're looking at here come from this portion of the shell right there. And what this is this is the shell in dorsal view. So you're looking down from the top. The head of the animal would have been here. The tail would have been here. So here you can see the shell in a living specimen. And this kind of almost boomerang-shaped bone that you see right at the front is known as the nuchal bone. And so this is one of these specimens here. This is a complete nuchal bone here. This is half of a nuchal bone. In trionychoid turtles, the nuchal bone is actually fairly systematically important. You can actually determine relationships from it. And what we have is actually two species of fairly large-bodied flapshell turtle in the Siwalik section, with this kind of morphology here. I'm sorry, actually, it's this one here. This morphology here actually showing up fairly late in the section, and so this appears to be a distinct species of Lysemes that occurred during the late Miocene. Other aquatic turtles, and the most common aquatic turtles are another group of softshell turtles. These are very large-bodied trionychoid turtles, represented by the shell fragments here. This is a shell fragment that can be assigned to the genus Chytra, which are the narrow-headed softshell turtles. And these are the largest, excuse me, of the South Asian terrestrial softshell turtles that you're going to find. They can have shell lengths that, including the soft parts of the shell, that can get to almost 1 meter in length. I mentioned two skulls as an undergraduate, and here they are right here, this specimen and this specimen. And these skulls belong to a very diverse, comparatively diverse clade of softshell turtles, known as the genus Milsonia. This is a specimen here. This is the Ganges softshell turtle. It's an extant species today, obviously. And these fossil skulls are actually very closely related to this particular lineage relative to other species. What's interesting about the geographic distribution of these animals is that this specimen here of Chytra has a fairly wide biogeographic distribution from India down into kind of the northern base of peninsular Thailand and Malaysia. And then these animals here are actually just limited to the drainages of the Indus, the Ganges, and the Brahmaputra and their tributaries. So these are limited to South Asia, kind of classically South Asia. We also have a diversity of what you might recognize as pond turtles. These are geomytid turtles. Some of them are, well, actually, they're all somewhat semi-aquatic. So these animals, of course, are very able swimmers and will do a lot of feeding in the water, but can also spend a fair amount of time on land. And they occur throughout the entire Siwalik section. We also get some very large turtles and some terrestrial adapted turtles as well, including both small-bodied and large-bodied tortoises. And so this is the plastron, kind of the backside of the underside of the shell of a small-bodied turtle that belongs to the genus Indotestudo, which is a living example you can see here. This is Aldebrachelles, the Aldebra Island tortoise. They don't occur in mainland Asia today. They certainly don't occur in India or Pakistan. But I don't have a photo of an equivalent giant tortoise because they're not there in India or Pakistan today. They were, however, there in the Miocene. And so what you're looking at here is the partial excavation of the shell of a giant tortoise. And this open space, this is actually kind of the inside of the shell, so the animal would have been upright. It's all broken here, but this is the belly portion of the shell. And in this space is where the right hind leg would have come out. Sorry, the left hind leg would have come out. So it would have been on the other side of the animal here. But what's preserved is his ventral margin. And this is Geological Survey of Pakistan officer Munir al-Haq prepping the specimen out of the ground for photograph. And this is a shell that's probably in excess of 1 meter in total length. And much like all Cenozoic giant tortoise specimens are treated when they're found in the ground, it was observed, photographed, and then buried because nobody wanted to try and collect an entire specimen of that. So we have lizards and snakes, we have turtles, and then of course we also have a fairly rich crocodile record. And the Siwalik crocodilian record actually shows a pattern of biotic change and turnover. There are 3 main groups present in the Potwar Plateau sequence. There's the lineage that includes modern crocodiles today, and so this is just a fragment of an upper jaw, and you can see kind of the large teeth exposed, so we have a large kind of caniform tooth. And this is one of the earliest representatives of the genus Crocodylus known globally. I think this specimen is about 12.4 million years, give or take, or maybe even slightly older. And then we have a couple of other taxa. We have gharials, the genus Gavialis, which is, of course, the elongate-nosed fishing specialist that is endemic to South Asia today and is critically endangered. I cannot convey enough how much the living species of this clade needs everybody's help. to keep from going extinct in fairly short order, and would encourage anyone listening to look up some of the gharial conservation projects that are out there today and consider helping out. So this is a skull that was found in the field in 1998. You can see these are the eye orbits here, the back of the skull, and that long narrow snout is slightly broken. And so gharials, gavialis, occurs throughout the Siwalik section. Crocodylus shows up around somewhere in, like I said, it's either 12.4 or 12.97. I'm very sleepy. And then we also have some very enigmatic, very large, and very interesting specimens as well. So this is the nearly complete skull of a very large-bodied tombstone. And tombstones are, today, is represented by a single species that lives in Borneo. They're known as false gharials. They have evolved a very similar snout to modern gharials. Now we actually think they may have inherited from a common ancestor. This is a fairly broad-snouted specimen. As you can see, the skull here is almost 1 meter long in total length. And we have several different skull fragments from this taxon in the Siwalik section on the Potwar that give it a range of just about kind of 14 to 13 million years, with some very large postcranial elements and associated osteoderms, the back armor, which suggests that these animals are persisting probably until about the transition from the Chinji Formation to the Nagri Formation. So that's kind of the rundown of the Siwalik reptile record and what we have, or at least what I was able to start to understand from 4 years of collecting on the Potwar Plateau, and also from so much of the collecting and hard work that the colleagues I work with on the Potwar had done for 20 plus years. And if we place this kind of in a larger framework, what can it tell us about environmental change, or what can it tell us about the relationship between environmental change and biodiversity? The reptile record is, as I mentioned before, it's very sparse compared to the mammal record, but it has an important story to tell that the mammal record doesn't directly tell us. And that is about aquatic habitats themselves. So despite the tremendous diversity of mammals in the Siwalik record, I don't think that there is an obligately aquatic mammal known. There may be some small-bodied carnivores that spend a lot of time in the water. I wouldn't be surprised if there were various otter-like critters. But I don't think we found a freshwater dolphin yet. But of course, there is a big diversity of aquatic animals or aquatic reptiles within the section. And so what you're looking at here is a generalized stratigraphic sequence of the Siwalik. And then what I've done is taken the kind of ratio of sandstone to siltstone in composite sections that Brian Willis published in 1993, and kind of accordioned it out to make the section actually correspond with what we know as to be kind of Chinji, Kamlial, and Nagri / Dokpatan boundaries. And so this should be taken with a grain of salt or a grain of sand, to be punny. But the point is just to show kind of the changes in the lithologic dominance of the river systems that Kay Berensmeyer talked about as well, where you're going from kind of coarse-grained, large-bodied main trunk systems in the Kamlial Formation to greater floodplain representation, greater representation of floodplains and overbank deposits in the Chinji Formation, with the resumption of big trunk river systems in the Nagri, and then of course this time transgressive transition from the Nagri into more variable depositional environments in the Dokpatan that also include a greater proportion of fine-grained sediments. And so that's kind of the generalized change in fluvial regime that we have through the Potwar. And what we see in both aquatic and terrestrial taxa in terms of how changes relate to that is we have a few patterns of first occurrence and last occurrence that, again, generally correspond to the changes we see. But I do want to point out that it's going to require a lot more actual sampling of the section to really robustly nail down definitive first to last occurrences. But one of the things I want to point out is that this large-bodied tombstone appears to be limited to the Chinji Formation. How far back it might be able to go with respect to the Kamlial is hard to say, because the Kamlial is comparatively poorly sampled. But we do find fossils of a very large-bodied crocodilian that probably corresponds to this animal through most of the Chinji Formation. We then get crocodylus showing up, and I think that is definitively at 12.4, possibly extended down to 12, almost to 13. And then crocodylus is unambiguously present throughout the Siwalik section, up here to about 6.35, which is the locality DP13, which was the youngest section that I sampled. But we know that since the Crocodylus was in the Punjab and in the Potwar until human history, we know that the taxon, of course, extends upward out of the Siwalik section. So we do have this kind of pattern of last occurrence of Tomistomens with first occurrence of Crocodylus. It's very tempting to wonder if these two events aren't actually directly related to each other and to start to wave arms about potential for faunal replacement or competitive exclusion. We don't have the record yet to do anything other than arm wave, but it is certainly an interesting observation to make and rife for additional sampling and study to determine. Also, of course, we do start to see first occurrences of various turtle taxa. The one that actually has a first occurrence and last occurrence pattern that is most robust in terms of sampling is the fact that we have an additional species of this aquatic flapshell turtle, the semis, which starts off at about 9.5 and then ends at about 6.5. And so we do see some patterns of occurrence and turnover in aquatic environments. In terrestrial habitats, we get large tortoises showing up kind of basically at part of the Nagri-Dokpatan transition. And then once again, they go up until the end of the sampled section. But we know that at least regionally, they had a record that goes up into the Pleistocene. We certainly see some big terrestrial turtle material coming into the older sections of the Siwalik group. For example, there are large turtles present in the Chinji Formation, but the very large animals don't appear to show up until once we start to get into the Dokpatans. And then the most kind of robust and well-documented pattern of change that we have is increasing diversity in snakes through the Siwalik section. And so what you're looking at here is the number of taxa and morphotypes, and this is some of the work I did for my dissertation, where you have standing richness, which is the actual number of taxa that we find at a particular interval. These are binned at 5 million year intervals, and then culled where poorly sampled 5 million year intervals were simply dropped. So that is the number of animals that we find, and then the dashed line is range through values. So where we find a fossil specimen at one interval, we don't find it at the next youngest interval, but then we find it again at an interval even younger. We just simply draw a line between them. So it's estimating the number by ranging through intervals where we might have some underrepresentation. And so what you can see is we get up to going from a low of 1 to 2 at some points, or actually, sorry, from 3 using range through methods in the Chinji Formation. We get up to 8 specimens once we get into the Dokpatan Formation. And most of this diversity are terrestrially adapted taxa. These are terrestrial snakes. And we find them both, this increase in diversity, both in surface collected records where people are walking along and picking stuff off the ground, but then we're also finding them in the screen wash records. And so I've mentioned screen washing a couple times and others have talked about it as well. Screen washing records are acquired through the washing and breaking down of bulk sediment that's collected in mass. And so, for example, this is from 2000, and these are bulk sections of sediment. I think this is from locality Y825, which I think is at the top of the Camliol, being loaded up onto Cody's. These bags of sediment are then brought into camp, where they are treated with water and occasionally kerosene to break down the sediment, and then sieved to produce vertebrate bone, primarily mammal microfossils, primarily rodent teeth. And that is a picture of a very young Will Downs with his screen washing setup, and I'm going to presume this is probably the early 1980s. So one of the things that I was interested in is looking at this increase in diversity and how that might be reflected in the screen washing versus surface collected records. And so Will had sent me some of the kind of what he would have probably have referred to as a bit of bycatch from the mammal teeth. And so this is just stuff that is accumulated at different very rich microvertebrate assemblages, showing the relative abundance of different types of non-mammals. And the thing to notice, of course, is that you have lots of indeterminate bone. Because this is sampling aquatic environments, you do have a lot of fish as the kind of aqua color running through it. But then you'll notice the dark green, how it increases dramatically once you get younger than about 10.5 million years. So here, here, and here. And that is, of course, the composition of the changes in relative abundance of small-bodied snakes represented by vertebrae in the record. And so we see in both the surface collected record, we're seeing an increase in snake diversity, but also in the screen wash record as well. So one of the questions I was interested in when this started to become apparent is what factors might actually be explaining or driving this increase in diversity. And so again, here's the changes in taxa or morphotypes. And by taxa, I mean organisms that can be identified to a genus or species name. Excuse me, and where I say morphotype, that is a distinct vertebral anatomy that is recognizable from the rest of the collection, but might not be able to be identified to a particular genus or species. And so we have that pattern as I described before, but then we can also actually look at changes in relative abundance of different snake groups through the section. And of course, we have fully aquatic Acrochordus, we have large body python, and then we have this diverse group of snakes that increases in their diversity, the colubroids. And as we get an increase in the number of taxa, we have an approximately concomitant, but not perfectly concomitant, increase in the relative abundance of specimens as well. And so you will remember I said Acrochordus is fully aquatic, and so you can see that through the Chinji Formation and through the Nagri Formation, Acrochordus is between 80 to upwards of 90% of the snake fauna that we recover. But once we shift from the Nagri Formation to the Dokpatan Formation, the relative abundance of Acrochordus drops. It does pick up here at this point to drop again. We are seeing a decrease in the overall relative abundance of Acrochordus in the fossil record as we transition out from the Nagri to the Dokpatan. Of course, what's interesting about that is that in terms of lithology, the Dokpatan is more similar to the Chinji than either of them are to the Nagri, at least in terms of kind of our classic understanding of the depositional environments of those formations. And yet, even though we have, again, this is a lot of overbank deposits and a lot of floodplain deposits and overbank and floodplain, Acrochordus is ubiquitous within the Chinji Formation, but not so much within the Dokpatan Formation. So it is possible that lithologic changes are actually, changes in fluvial architecture are simply driving preservation differences, or it's actually changes in fluvial architecture and changes in regional environment that are actually resulting in the changes that we see that are biotic. In looking at the diversity of snakes, one of the things I wanted to know is whether or not this was comparable to changes that we see in, for example, mammal microvertebrates or in the rodents. And fortunately, that work has been done in terms of species diversity, originally by John and everybody in 1995. And then Larry Flynn and company did re-examine this in 1998. And what you can see in terms of species diversity of rodents, the dashed line, is that rodent diversity is actually highest kind of close to 14 million years in the Middle Miocene, and then kind of has a bit of a decline continuously through the Siwalik section leading towards the top of the exposed Potwar region. Or I should say the top of the Dokpatan around DP13. And this is exactly not what you would expect if the pattern that we were seeing in the snake record was simply a preservational artifact. You might expect these two records to actually trend with each other. And instead, what you have is snake diversity appears to be increasing while rodent diversity is decreasing. Again, that's interesting, not just because it suggests that there's not a kind of common mechanism controlling this diversity, but also these animals share ecosystems, and snakes are predators of rodents. And if it was something like prey diversity that was driving snake diversity, you might actually expect to see a positive relationship between them, and we don't. So I'm actually going to jump back real hard just to talk just a second or two more about this. My conclusion from the work on snake diversity is that what we were seeing is the increase in overall diversity is because we were getting kind of greater environmental heterogeneity proximal to depositional environments in the Dokpatan Formation. The kind of larger scale or continental scale diversification of derived snakes actually predates by some considerable chunk of time the increase in diversity that we see in the Siwalik Group. So I think it's more likely that what you have are fairly diverse derived snake communities that are distributed throughout South Asia, but their ability to become entrained into the Siwalik depositional system really depends on having environments that are favoring basically the environmental heterogeneity that's going to allow for an increase in taxonomic diversity, and that's going to be proximal to them getting included into the fossil record. And as a component of that is you're losing kind of the large-scale fluvial system, or at least the fluvial deposition or dominance of aquatic taxa concomitant with an increase in diversity in terrestrial taxa. So one of the things I've been interested in for a very long time in thinking about reptiles as poikilotherms is how they might be able to give us some bit of predictable relationship or predictable understanding of environment or climate that we could potentially use to actually reconstruct or understand the past to some precision. And I've been working with various colleagues through my career to kind of come up with different models for that. And I'm working right now with Professor Michelle Lawing at Texas A&M University on looking at the climate parameters across a diversity of living reptile communities to understand the kind of variability that you see within these communities and how you could actually apply this to fossil communities to understand both standing environments and how environments change over time. For two examples of this, or for an example of this, I should say, we have basically the mean annual temperature and the annual precipitation within the range. So this is the frequency distribution of those two variables within the range of the genus Acrochordus today, and also within the range of living Thomistoma schlegeli today in Borneo. And I picked these two animals, obviously, because they're both quite common in the this is quite common everywhere. This is never common, but they both occur in the Chinji Formation. And so what I was interested in looking at is how the environmental parameters of their modern habitats might be similar or different to each other, and how those similarities or differences might tell us something about how they would respond to environmental change. And the thing I want to point out is that in terms of mean annual temperature, kind of the optimal temperature in terms of frequency distribution for these animals is almost identical. They both have a fairly narrow range of temperatures within which they have high geographic ranges or diversity. But what you'll notice is that the precipitation, the range of precipitation, annual precipitation values within their ranges is actually quite different. So Thomistoma is fairly restricted to the kind of wet habitats of equatorial Borneo and southern peninsular Malaysia today, whereas Acrochordus certainly occurs in these wet habitats as well, but has almost a bimodal distribution where one of the species is actually in areas that are actually highly seasonal in terms of precipitation variation. And actually, it looks like this animal can inhabit environments that become seasonally dry, so long as there is probably at least some additional source of water that is providing aquatic habitats. Why might that be interesting? Of course, one of the things that we see through the Siwalik section is that this kind of wet habitat specialist like Thomistoma is gone for sure by about 12 million years. And again, it would be interesting to know what the real last occurrence point for that animal is. Whereas Acrochordus is present at least through the Miocene section. And if we look at some of the climatic changes that we see represented by oxygen, obviously by about 10 million years, by about 10 million years is very good evidence for seasonal evaporation. We're looking at seasonality in the inception of monsoons. But it's also possible that we might actually have some seasonality slightly earlier. And if we do get earlier seasonality, this limited range here might explain why we lose Thomistoma fairly early on, but yet we're keeping Acrochordus. And that would be due to basically changing climatic environments that would allow for an animal that had a wide range of tolerances for annual precipitation to remain while a wet climate specialist would be extirpated. And so, again, what we're doing is kind of looking at the modern world to see how this can inform on the past. But we're also interested actually in flipping the script a little bit. And so on the right is from a study I did some years ago with some colleagues to actually use the fossil record of reptiles as a paleothermometer, if you will. And we were doing this because of the discovery of a very large snake from the Paleocene. And what we did is we used the known relationship between metabolic rate and body size in living snakes, which is described by these two curves here. We use that known relationship to actually predict the ambient temperature within which a larger snake would require to actually have successful metabolism and to be able to survive. And by using this metabolic paleothermometer, as we called it, we were able to reconstruct some of the past climates in the Paleogene greenhouse tropics. If we were to apply this same method today to the Siwalik record, where Acrochordus is twice the size as it is living, we would get unreasonably and ridiculously hot temperatures for South Asia in the Miocene, well pushing 30 degrees or 30 degrees plus mean annual temperature, which they just simply weren't. For a whole wide variety of reasons. There are many proxies that indicate that you could not achieve these temperatures. But this model here requires a couple of things that are important. One, for the comparative approach, the extant taxon has to have a maximum body size for its given temperature. In other words, if this snake here were smaller than it could possibly achieve for its given climate, then it would give us an erroneous result. So what we can do is we can actually take the kind of mismatch or the offset that the relationship between modern taxa gives us for past climates. We can actually flip that and say, if this model, this predictable metabolic relationship to climate model is inaccurate, what is that telling us about the modern world? And what's important and interesting about that is, in theory, we can then actually develop the past to inform on the present. And of course, one of the things we know about the present is that the modern world, especially for vertebrates, is fairly interrupted by human behavior and has been for a lot longer than we realize. And we can actually start to begin to use the fossil record to address questions about the modern world as opposed to the other way around. And so finally, just to kind of end this and get back to what I'd started with originally, which is talking about the evolution of some of these biotic provinces. What we can see with the Siwalik Group is that we actually have some fairly early, at least early in terms of the Potwar record, by about 14 million years, we have representatives that are kind of part of the modern endemic South Asian drainage systems today. We have soft-shelled turtles, we have Gavialis, we have some of the freshwater turtles. Then we have a mix of taxa that are more characteristic of the Indo-Malayan province, such as python and bungaris, and then the large soft-shelled turtle, Chitra. We have taxa that basically span the entire geographic range of Southern Asia to Australia. So Acrochordus, which occurs all the way through this range here, up again until the kind of southern part of Southwest Asia today. And then we have representatives of just the oceanic component of insular greater Sunda, such as Temistamens. And then what I think is most interesting is that we actually have components of what is the insular Sahel platform of Australia today. And so what this kind of range of taxa shows us is that the kind of some of the modern breakdowns that we have, some of the provinciality that we see, is fairly recent in terms of its origination. So certainly we know that a lot of the kind of provinciality that we see in oceanic Southeast Asia is the result of flooding of what was previously a contiguous platform with the mainland of the continent, and that happened fairly early on, or fairly recently in the Pleistocene. But certainly the geographic isolation of New Guinea, I should say, and Papua New Guinea, as well as Australia, from the mainland, or from the larger Asian components of this contiguous area, is much more poorly constrained for a lot of taxa. And the fact that we're finding something like a pig-nosed turtle so many thousands of miles away and fairly young into the section suggests that a lot of what we get in how we think about this modern provinciality is going to be a fairly recent event. That might seem intuitive to some of you that specialize in thinking about some of these areas, but actually there have been hypotheses that, for example, Lydeker's line and Wallace's lines may have been established as early as 20 million years, and sometimes some have even argued based on molecular data even into the Mesozoic. And we certainly don't have evidence to support that when we look at animals such as these pig-nosed turtles, which of course are occurring in South Asia, very far away from this region of the world, as young as 7 million years. And so I finally want to end this by reiterating something that both John and Catherine said in their talks when they were thinking about out loud about what they would love to be able to do in terms of going back and trying to address questions that they have and think about what they could do in the future. And I will take a backseat to nobody wanting to get back to the Siwalik to understand what is going on with the reptile faunas. I was able to get, like I said, enough out of material for my dissertation to begin to ask some of these questions. And 21 years after my dissertation work, I think I actually understand the animals well enough to start to intelligently frame those questions. But if I knew then what I know now, I would do things very differently and examine things differently and collect things differently. But one of the things I would be desperate to do is to look up section and to get into this area here. And I have to say that in my youth, I was more interested in going down to where I had presumed things would be weirder and different than today, without realizing that actually some of the most strange and non-analog Siwalik taxa are actually very high up in the section, such as some of the really incredibly giant reptiles from Chandigarh in the Pliocene. And I would love to see what the Potwar section looks like as we get higher up. And of course, that's problematic, as Kay's talk indicated. The kind of quality, the density of the fossil record decreases greatly, but it would still be great to get back there and to get into the rock and to start to see what we can find. And I know that there are probably some students out there listening, and what I would in Pakistan, and what I would say is, that is something to be thinking about, is if you're thinking about projects and things to do, looking at the reptiles, and especially looking as you can kind of, if you can march from the Miocene up into the modern, through that section, you're going to see some really interesting changes. Most notably, things like Acrochordus leaving this portion of South Asia completely, changes to the pattern of diversity in things like crocodiles and gavials and turtles, and then some of these weird, like I said, some of these very weird, very large animals from North India, whether or not they're making it into kind of across the 300 plus kilometers over into the Punjab, or in the Pakistani part of the Punjab. So to conclude, basically, I hope I haven't rambled too much in my sleep-deprived state. Just to say that the Siwalk Group it's very taxonomically diverse. It has a wide range of representation of South Asian to Australian taxa from these provinces. It is, as I said, it's very sparse compared to the mammal record, but we do see compositional changes that are consistent with environmental changes. But we really need more material. Like I said, and I'm just repeating myself. I was able to get enough information and from my collection, and then from all the work that all the other Siwalophiles had done, to start to frame these questions, to get a general sense of where taxa may be occurring, and to really actually get a better handle on these patterns, to get a more precise biostratigraphy for these reptiles, is just going to simply take more sampling. So I will stop there, and I will simply thank all of these people who have been so influential to me and so helpful and so tolerant through the 2 decades of my work on and off with the Siwalak Group. I have worked in the Neogene and in the Paleogene and the Cretaceous pretty much around the world. I am addicted to international fieldwork and travel, and I am addicted to international fieldwork and travel because of falling in love with Pakistan when I first got off the plane in 1996. And so to everyone here, I will say shukriya, and shukriya for your attention, and I will be happy to answer any questions that anyone has.
Very nice, Jason. Thank you for a wonderful talk, and astonishing to see the biogeography of things like snakes. I mean, hard to imagine the capacity of those creatures to cover those distances, and fascinating to think about. And I think we've got some time for some questions. Perhaps if anybody has a question, they might want to speak up. Mukhtar,
yes, they can use the raise hand option so that we can take them into line. Yes, we have Jukar.
Hey Jason, so thanks for a great talk. You don't hear a lot about Siwalik reptiles, so that was quite eye-opening for me as someone who works in the young part of the section. The question that I had was, do you find equivalent faunas in parts of the world like East Africa, where you do have a lot of faunal similarity when it comes to the mammals? Do you have a lot of similarity when it comes to the herps as well, or is that largely with South and Southeast Asia?
It is, so there certainly, if you're talking about kind of connectivities between Africa and Southeast Asia, there are some, for example, the flapshell turtles that I mentioned. The technical name for them are cyclanborines. They are very widely dispersed through sub-Saharan Africa today and then into South Asia. But there are a few things. So there are fossil gharials that were in East Africa for a bit. But yeah, and so I guess now that I'm running through this, the answer is yes and no. So you do have some kind of ubiquitous attacks at pythonids, for example. Pythons are across the old world. Gharials, again, had a wider distribution in the past. But things like Acrochordus and a lot of the more kind of exclusive turtles at exclusive taxonomic levels are pretty segregated. We do actually see kind of a greater provinciality among the reptiles than you see among the mammals.
Cool, thanks.
And just to jump on that, also a lot of the patterns of faunal interchange between Asia and Africa, they're very well constrained especially for megafauna. It looks like, especially some of the work that people like Nancy Stevens and Pat O'Connor have done in the late Oligocene of Tanzania, they're actually finding that especially squamates, lizards, and snakes are going over from Asia into Africa much earlier.
Catherine Wedley, please unmute yourself. Your mic is muted. Please unmute yourself.
You might have to give her permission to unmute herself.
I think there is some problem, so we will allow Paramjit and we'll come after them. Paramjit, please unmute yourself.
I think Paramjit
Can
is.
you audible?
Yes, Paramjit, now you can ask.
The presentation was quite interesting, and I really enjoyed it. I have two questions. The first questions, I just want to know the migration pattern of baranid between Asia, Africa, and Europe. In Africa, the oldest record was reported from Miocene by Holmes et al. 2010. In India, there was also a record of all this baranid from middle part of lower Oligocene. So my question is that what is the possible migration of baranid to India, whether it was from Europe or Africa or Southeast Asia? And the second question is, there are sometimes difficult to identify between the boidae and the pythons. What are the important characteristics to identify the boidae and the pythony? Is it palaeoclimates foraminifers important to identify boidae or pythony?
Yeah, first of those are two fantastic questions. I do love getting paleo reptile questions. So to answer the first question, the early history of Varanus is, as you can imagine, kind of confused and not well understood. So the African radiation of living Varanus is actually endemic to Africa. And then that is the kind of sister taxon, if you will, to the Asian group. When that split occurs is pretty poorly constrained. So proper Varanus, where we would, not a varanid, but part of the genus Varanus that can be related to the African taxa, the oldest record for that can be placed into a phylogeny is about 20 million. You're absolutely right. There is a Oligocene material from Asia. There's even late Eocene material from North Africa, which looks very much varanid. But whether either of those is actually within the living group of Varanus or is outside of the living group of Varanus, we don't yet know. So, I think that the split between them is at least earliest Miocene and probably late Oligocene. But to really get down to that point, one of the things that we would need is a very good African Oligocene record. And as people who work in Africa will tell you, those are not common. Sorry, for your second question about pythons and boids. So yes, there's a series of characteristics for boids. They will often have a set of two small foramina on either side of the cotyle, the paracotylar foramina. this is going to be some, I guess the expression inside baseball is geographically limited, but the very kind of fine-grained differences. Pythonids have a tendency to have very, there is a ridge between the pre and post zygapophyses called the interzygapophyseal ridge, and in pythons that's more well-developed often that you see in a lot of boids.
Okay, thank you.
Mm-hmm. It looks like Catherine is still muted. Oh, there she is.
Yes, yes.
Can you hear me now?
She's out.
Yep.
Thank you. You worked your magic behind the scenes, Mukhtar. Thank you. Thanks,
Thank you.
Jason. You've done a marvelous job with a challenging record. Let's put it that way. I have a question that you may have hinted at as you were talking. Do any of the aquatic reptiles give us an indication of the magnitude of the rivers? I'm thinking of how wide the channels were, you know, how deep they were, how you've talked a little bit about potential seasonality. And I'm just wondering if there's a relationship, as there sometimes is with fish, between, you know, body size and river, you know, environment size.
So that's a great question, Catherine. And it's one I was very interested in trying to get to in the dissertation. And so again, we do see in what we presume to be there's kind of two parts about it, which is that where you get, of course, the really big systems in the Cameleon and the Nagri, you don't have that much of a record. But where we think we do have larger systems in the Chinji Formation than in the Doak Baton, we do get these big temistamines, and these animals are really big animals that are in the Chinji, but not in the Doak Baton. I looked for a signal in body size and Acrochordus as part of my dissertation and didn't really find one. And I haven't seen, you know, I'm not a fish expert, but I've certainly seen a lot of the fishes from the Siwalak Group, and I didn't actually notice a particularly big difference between, and you might have better knowledge of that than I do, a size difference in the fishes from Chinji to Doak Baton, for example. So the long answer is I haven't seen definitive evidence that you can kind of see a big, large-scale change in the aquatic fauna with respect to changes in fluvial architecture, at least between those two. That being said, and this is important, the way that I was measuring body size and Acrochordus in my dissertation is not the way I would do it today. And so that regression equation I showed in my talk would be, I think, very useful to apply to the record that we have, and it's something I actually really would like to get back to doing.
Great. I hope we all get back there.
Yes.
So we have another question from Kay. Please unmute yourself.
Okay. Yeah, actually, I have two questions now. Following on Catherine's, is there evidence from modern fluvial systems for a correlation of body size of the reptiles with the channel parameters? So that's my first question. Then yes. I'll wait till you answer, and then I'll ask my second.
So the answer to that is, I think, mostly apocryphal. I'm not aware of any study that has actually sat down to kind of look at something like depth or channel diameter to say the size of large or small reptile. There are some studies that look at habitat area and body size, which definitely show that larger body size animals have larger habitat areas, but that has a tendency, if you're thinking about crocodilians, that's usually done as kind of a, if you will, a sphere as opposed to the parameters of the aquatic system. It would not be impossible to get those data, however, to see if there is a correlation.
Okay, thanks. And the other question is, you showed the graph of the assemblages that Will Downs gave you to look at, and you know, there are patterns in there that may or may not indicate changes in the relative abundance, because we don't really understand how those accumulations that were screenwashed occurred. And they may have, I mean, all those small snakes may indicate that there was a particular predator involved in the initial formation, even though these are slightly reworked into fluvial deposits.
So I looked at that when I did the initial study, because of course the paper in Doc's volume had actually looked at the relationship between grain size and diversity and concluded that it had to be a biotic accumulation agent for the mammal screen washing records. And we running the same analysis on the snakes, you basically shows kind of the same thing, but then you also have lots of things like fish and crocodilian, and so there's probably, my assumption is that there's probably multiple agents of accumulation. And yes, the question is the increase in snakes because something is now eating them that wasn't eating them before, or are they simply being entrained into the system along with things like bits of crocodile and bits of fish and amphibian? And the answer is I can't, with the data I have, disentangle them from each other, but we definitely do see a change. And to double back on that, however, I do want to point out when I'm talking about this increase in diversity, I do think it's because of environmental changes proximal to the system in the Doak-Baton Formation. And I suppose having a predator that's now actually sampling that environment isn't necessarily completely different from having the local environment being the driver for diversity.
Yeah, and I could add that in my study in modern ecosystem in Africa, there are multiple agents that accumulate small bones under trees, so there are catfish and lizards and snakes and bird bones and mammal bones that can build up under particular roosting sites and you know this well. I mean, it's been documented in other contexts too. So I suspect that is playing a role here.
Oh yeah, absolutely.
Thanks.
Thank you.
Good. Well, let's see. Is there one more question there?
Yes.
Well, it's more a comment than a question. So you said that there was no record of marine mammals in the Siwalik. So in 1876, Lydecker reports a tympanic from an alleged platanistoid from Theobald's collections. Now he showed it to William Flower at the NHM, and Bill Flower didn't think it was a cetacean tympanic. He thought it was an ungulate. But I have no idea what happened to this specimen. It's just twice. But I've got the papers if anyone wants them.
That's excellent. I would have thought that given the intensity of collecting on the Potwar, that if there were dolphins, the dolphins would have been found. But you never know. The big Temistamine could have been eating them all.
Just a quick comment. There are very, very rare ray teeth, suggesting that there are slight marine elements that make it into the rivers, and some of them are even Doak-Baton in age.
Wow.
That's what I've learned by reviewing all the fish data.
So that's interesting because that is very far inland to, I mean, we're well beyond anything you could call estuarine by 1,000 kilometers at that point. So that's really interesting that you can get salt tolerant taxa that far upstream.
But you get rays in Lake Turkana, so.
True, but I thought the argument was that there at some point there's a direct drainage straight into the lake, and it wasn't nearly as far away from the coast as the Potwar would have been.
Yeah, but that's open to some debate too.
Yeah, fair enough.
Apparently these rays, these are the dasyetid rays. Apparently some of them make it quite far up. Okay. Some of the big rivers of South Asia as well.
That's amazing.
Mm. Good for them.
Yeah. All right. Well, I think we've had a wonderful evening and morning, and I think I've seen the sun rise in England. I want to thank everybody for participating, and of course, especially to thank Jason for his presentation. And I think it's a little bit of an eye opener to consider a faunal group that's somewhat inconspicuous, and the significance that can be derived from it. So a wonderful presentation and lots of food for thought, and also opportunities for further research. So again, I want to thank everybody